word, and I request to the chairperson of this session, Dr. Developnt. Please come on the dais. And our resource person, I request Dr. Avinash sir, Department of Botany, Savitribai Phule, Pune University, Pune. Sir, please come on the dais.
Session attendance पण घ्यायचं. सकाळची
Till then, I request to the Professor S.N. Shinde sir, Department of Botany, to award a bouquet to our chairperson. Thank you, sir. RSML, please have your seat on the dais.
Photographer put together. कसं काय? Photographer.
Dr. Avinash Piyane, an esteemed Professor Savitribai Phule, Pune University, for his contribution in plant biotechnology and the environmental sciences. Now, this session will start with the enthusiasm. Students and the fellows, I think you are very enthusiastic. Are you? Do you get your energy again? Yes, very good. Now I request Professor S.N. Shinde sir, Head Department of Botany and the convener of this program, to offer a bouquet to our chairperson, Dr. Developnt sir. Please give us our applause. Thank you so much, sir. Now again I request to Professor S.N. Shinde sir to welcome for this resource person for this session, Dr. Avinash Piyane sir. Thank you so much, sir. Now I request to Mr. D.R. Award sir to introduce our resource person.
Sushethropati Shikshan Sanstha, Rajashri Shaw Mahavidyalaya, Autonomous. Department of Botany organizes yearly sponsored one-day national seminar on advanced research and development in life sciences. I am here to introduce this technical session resource person, unique, dynamic, energetic. Teaching is passion and research is a mission. Considering person, renowned ecologist, Professor Avinash Ade, Senior Professor, Department of Botany, Savitribai Phule, Pune University, Pune. Sir completed his BSc from Amravati University, Amravati, as well as PG with gold medal. Sir completed PhD in Dr. Developnt University. Sir has more than 26 years teaching experience as well as research experience. Sir working as a research guide under the supervision of Ade sir. 13 students are postdoc, PhD completed. Running 5 students working under his supervision. He also guided 5 MPhil students and more than 60 PG project students, master degree projects. Sir has already I know the research mission is the research. So sir has a lot of publication. I am not talking about all this, but just little bit and more notable publications I mentioned here. The research paper in Science Citation Index more than 20. Communicative impact factor is 76. Impact factor. Average impact factor is 4. Sir has Google Scholar Index 17. HI Index 26. Sir publishes non-science citation index research papers 35 and others. Sir is a writer also. Sir has writes 11 different books in sciences. Most core valuable subject is ecology. Sir has particular workshops, attended different workshops, completed workshops, seminars. 35. Sir presented paper in different conferences and workshops. 45. Invited conferences in 13. And he is a member of Indian Society of Genetics and Plant Breeding. Also plant pathologist and ecology society. Sir, the research area of sir is the bioremediation and microbes interactions. So this bioremediation is very much important because of everything is biodegradable, but something that cannot be degraded biologically, it means naturally. So sir has given idea about that how they cannot be degraded or disintegrated naturally, and they have searched to ecologically, economically, or eco-friendly, how they can work with the help of microorganisms to reduce the pollutions in the area of plastic waste management, oil degradation, pesticide degradation, bio-extraction methods, and also plant microbes interaction. We are the science students, we know the interactions, different kind of interactions, beneficial, harmful, disease interactions like the rhizospheric, ear microbiota, means those microorganisms they can be survive within the microscopic area that beneficial for the growth and development of the plants to enhance the defense mechanism of the plant with the by specifically biological control and inorganic conjunct. It is much important to enhance the defense mechanism in a plant pathology or in a plants breeding. So with this brief introduction, I am not taking much more time with you and today's research person. Thank you.
Thank you, our sir, for such a nice and brief introduction about our resource person. Now, this is the time to welcome on the dais and the eminent personality, Dr. Avinash K. sir from Savitribai Phule University, Pune, and I request him to enlighten us with his thoughts. Please give a huge round of applause for him.
Hello. Good afternoon all. Especially afternoon session is quite lazy because of the lunch, and after lunch our digestive system becomes so much active, and that suppresses other systems which are activated for research. Anyway, I am not allowing you to sleep in this session. Honorable Chairman of this particular session, Dr. RSML sir, Honorable Convener of this Professor Shinde sir, Organizing Secretary Dr. Kalyan Saud sir, dignitaries of the dais, I can see my students before me. So I am taking this pill of the lecture in MSc class. So with the introduction, Dr. Janoba Awad sir has told about my special interest, and he has described that I am an ecologist. So what is the meaning of this ecology? Ecology I have chosen purposefully because it is having a lot of freedom. You can jump on one to another and anywhere, and all these are accommodated as interdisciplinary science that is known as ecology. So just like that, this subject is going to touch the global phenomenon, similarly local phenomenon, then it will be touching to the molecular phenomenon, as well as biochemistry, as well as you can say other plant physiology, plant functioning, etc. So this particular topic has been selected for this talk. So that is about the plastic waste management. So all of us know that the plastic is one of the pollutants, and it is difficult to manage the plastic waste, and that's why there are so many rules and regulations by government, both state as well as central. Time to time these are getting changed, and just like you can say Mumbai Motor Vahan Kaida, amuk, according to that, we don't follow at all the rules and regulations of the traffic. Just like that, we also ignoring the rules which are set by the government regarding the usage of plastic. So such plastic, if you want to understand, so basically we have to ask the question, what's the matter? So plastic is a matter, of course, and this matter is just like any other matter which is existed on this earth. So matter we generally see in three states. These are reported as solid, liquid, and gases. And fourth state is also there, that is known as plasma state. So this is the involvement of the physics, of course. And chemically, if you see, the matter is composed of elements, then mixtures and compounds. So these basic things are there. And if this is the nature of the matter, then what happens with the matter and how this matter is getting regulated on this earth, and who is regulating this matter on this earth, that we have to understand. Next, please. So, who is going to regulate the matter on this earth? If we ask this question, then answer is energy. In Hindu mythology, there is a concept of Shiva Shakti. So Shiva means the matter, it's the equivalent to matter, and Shakti means the energy, power, that we know. So Shiva Shakti or Ardhanari Nateshwar, that concept has been focused. So it means that this is a combination, or very nice combination of the matter with the energy. So matter is regulating, energy is regulating the matter, and according to the Einstein equation, E is equal to mc square. So E is the total energy of the universe. C square is the square of the velocity of light. Okay, and m is very much negligible. So matter, when it is tending towards energy, its mass or its quantity is negligible, and it is now converted into tremendous energy. So E, which is the total energy of the universe, and C square, if you compare, then these are near about same. So when the matter will be converted into energy, or when the energy will be converted into matter, these are, this line of demarcation, these are very much faint. And in view of that, we can say that the matter which existed, this matter will be the conservation of the whole matter which is present on this earth. Similarly, energy is also existed. Once it is obtained from the sun, then it is going to regulate the life activities, not life activities, non-living activities are also regulated by the energy. So this particular concept has been taken here, and due to this, we are coming to the global phenomenon or universal phenomenon, which to explore for our core topic, that is plastic. So suppose the plastic is basically a particular material which is a polymer, and that polymer, if you want to degrade or if you want to dispose, then what should be the strategies? These are to be degraded. So for this degradation mechanism, of course, this is our product, human product, and that's why we have to find out the solution. So if we are finding out the solution, we have to understand the global phenomenon. We have to consider it, and according to that global phenomenon, we have to give the touch of this global phenomenon to this particular task. So while considering globally, you will find that there is a cycling of the matter. So biogeochemical cycle, a very typical example, carbon cycle, nitrogen cycle, or sulfur cycle, phosphorus cycle, these are there. So just like that, there are n number of elements, probably out of the discovery, according to the Mostley's long form of periodic table, 118 elements are there. So for each of these elements, this cyclic movement is possible. And same, we can find out for the compounds, similarly, uh, for the compound also, because compounds are basically made of the elements, and for okay, yeah.
दिसतंय का आता बाकीचे ना?
I think this corner.
आता दिसतंय.
So compound is also following this particular rule. Now, the polymers or polythene or plastic, these are basically compound formed due to the usage of elements. So elements when combined together, elements get combined together because of the valences they have. Valences are technically defined as the combining capacity. So this combination is there, and because of this combination, we can produce wonder molecules, and plastic is one of them. So plastic is prepared from the petrochemicals, and this is wonderful molecule human has ever made. Next. So this is the diagram of plastic, how this is getting generated. So plastic is basically a polymer, and any polymer, if you find its composition, then this is composed of monomers and dimers. If one molecule is there, monomer, two molecule dimer, three molecule trimer, like this, polymers which is the term, and n number of times if these molecules are repeated, then this will be a polymer. So such polymers are derived. Next. So based on this polymer, the properties of the plastic have been defined. So what are the possible properties that we can understand? These are the lightweight compounds. The weight of a polythene bag, if you consider, then it is as good as nothing. Okay, but it has very good strength, which can carry out heavy loaded material. Okay, so actually, the polythene bag which is available in the market, that is of say 50 micron as per the government rules, but even less than that, 25 micron polythene bags are there, and this 25 thickness, 25 micron thickness of polythene bag can carry 2 kg of grocery. Okay, so this much is considered as very much strong. This is described as tensile strength. So tensile strength of the polymer is too much, and that's why durability is too much. So if the durability is too much, then obviously it will be difficult for getting degraded, or it will be difficult for getting destroyed. And that's why on one side we are considering polymers are wonderful molecules, and these are the options for so many things. Say for example, a cloth bag. Cloth bag you can carry for a vegetable cooking, or pores of the clothes. The circulation of the air will be okay. But what about ink? What about the eye? So for this, the polythene is ultimate solution, because you can't carry milk in a cloth bag, or any other jute bag or any fiber. It is of no use except polythene. So that's why I am calling it as a wonder polymer. Okay. So this polymer was discovered by a human being, and then human had changed his life. These are called as revolutionary changes. Okay. So bakelite was first plastic type that was discovered, and later on so many polymers, so many plastic derived substances they were made, and we are now positioned today that our life without polythene is still quite difficult. So right from the morning up to the sleeping time, everything, every step, every event, we have to utilize the polythene. We have to make use of the polythene. And because of this enormous importance of the polythene in our life, the production of the polythene or production of plastic has been increased too much. Negative. So in earlier picture, we have seen different material which is made up of plastic, and we have to understand the basic types of the plastic. So plastic is classified into two major categories: the thermoplastic and thermoset plastic. So thermoplastic, they are very typical in structure: polythene, polyvinyl chloride, polystyrene, or polyesters, acrylics, and nylon, etc. And thermoset plastic, that is polyurethane, fiber-reinforced plastic, acrylic, polyester resins, and vulcanized rubber. So these are there. So what is basic difference between this thermoplastic and thermoset plastic? We have to understand. Okay. So here, for confirming the property, confirming the type of the plastic, we have to this plastic pieces on the frying pan, and then you will see that left-handed figure is showing melting immediately, while right-handed figure that is showing portion or corners are only melting and other material is still intact. So this is the basic difference. One is melting, why? Because of the simplicity of the structure. So these are the chain structures. These chains are independently associated, and because of this independent association, due to the sliding mechanism of chains, the molding of the plastic becomes easier. In the right side of diagram, you can see the chains are there, but these are firmly attached with each other. So this firm attachment of the chain makes it difficult to mold, and that's why this is known as thermoset plastic. Next. So these are the examples of thermoplastic. You know the names of these things, so I need not to tell you. Next. And these are the thermosetting plastics. So thermosetting plastic are utilized in very complicated things. For example, this is the aircraft engine. Example, that is vulcanized rubber. Okay. So rubber is a hydrocarbon derived from the rubber plant that you know. Okay. Avia Brazil is the name of rubber tree. So from this extract you can take, and rubber as a polymer you can prepare. But this rubber is quite less in strength, and due to addition of this crossing substances or interlinking substances of the rubber chain, the vulcanization is carried out, and this interlinking is established because of the addition of sulfur. So rubber plus sulfur, vulcanization is done, and vulcanized rubber is used for making tires. Okay. So such tires capacity, if you see, then you will find that surprisingly it can hold the air, and air you can measure in terms of I can say particular tire pressure. So tire pressure about 30 to 35 that is maintained in every wheel of the vehicle. So this particular pressure keeps the air which is encapsulated in that particular tube, and the tube is encapsulated by the tire. There are certain tubeless tires also. So this entire air is held, and this holding of air is possible because of this wonder polymer. Otherwise, instead of that, we would have used the wheels of wooden material or wheels of certain metals. They are of no use. So tires are ideal wheel, and that's why these are being used. So for tolerating the strength of this load, the tire are found to be very much useful, and it is said that 85% contribution of the air which is encapsulated in the tire, inflated tire, that is there to carry the load and 15% contribution is there for the bearing of this load, that is contributed by rubber. So with 85% and 15% is 100% contribution is made, and this rubber tire is now able to carry so much heavy load. So you can conceptualize this carriage in the form of a long truck, and that truck is carrying the railway engine. So you can consider what is the load of railway engine, and that is easily carried from one place to another place by a truck, and this truck is having wheels of rubber. Okay, so such thing is there, and that's why right from the beginning that wonder polymer, plastic is a wonder polymer. So according to the uses, the types of the plastics have been designated, and these are categorized, and these are even mentioned on the plastic material. So one corresponds with the polyethylene terephthalate. Second corresponds with the high density polyethylene. Third is vinyl polymer. Fourth is LDP, low density polyethylene. Fifth is polypropylene. Sixth is polystyrene, and seventh is miscellaneous. So all these polymers are being utilized for the making of objects which are in our daily usage. Next, please. So because of this particular fact, the plastic has been utilized in every field which is related to human being, and most of it is taken up by the packaging field. So every kind of packaging, if you see, then plastic is ideal material. So it may laminate your degree certificates. It may be utilized here for the carriage of vegetables, carriage of groceries, and even in sun or rain, nothing will be happen to that. Even nowadays, people have invented certain bags for carrying the mobile also. So even in a heavy rain, this mobile is protected, which is a highly sensitive instrument, and many more high-end equipment. These are also packaged by the plastic. So this is being utilized especially more in the packaging industries, and followed by packaging. Other sectors are there, that may be in building and construction, electrical and electronic equipment, or furniture, houseware, transport, agriculture, then toys, mechanical engineering, and other miscellaneous products are made from the plastic. Next, please. So due to this ideal material, the production of the plastic has been increased, and it is tremendously increased. Means at the discovery of this long step of curve you can find, and as we are understanding the drawbacks of the plastic, then the progress of the production has been somewhat mild. But there is no negative drop down of this production. The production is increased positively as the time gets passed. Next. So due to this particular situation, the pollution will be obviously there, because on one side we are looking for the durability, the strength, and because of this particular property, the disposal will become difficult. And if this balance is not maintained, then obviously the accumulation of plastic will be there. So plastic pollution will be the phenomenon that we should observe. So such plastic pollution, if it is to be observed, or if it is to be find out, then very good site will be the sea coast. Okay, because whatever the plastic we are using and we are throwing it in the garbage, that plastic get collected into the water streams, and these water streams are mixed with the river water, and rivers are depositing this plastic into the oceans. And here, just like any material, any solution we are shaking in the container or pot, just like that, the ocean shake this, and after shaking this will be thrown away, and this thrown plastic will be there on the seashore. Okay. Out of that, The heavy loaded plastic may be settled down at the bottom, and some other middle type of plastic that may be suspended in the oceanic water. So everywhere you will find a plastic. So if you want to look for the plastic in ocean, then you can find out the surface. You can find out the middle portion which is in the form of suspended, and you can find out the settled down plastic also. So for all the animals or all the biota which is existed on the ocean, this will be having the problem of plastic exposure. Next, please. So these are the impacts of plastic. So this bird, although it is not aquatic, it is present on the ground, but due to the carry bag, polythene carry bag, which are spreaded by the wind current, due to this, the bird is getting suffocated. This cow is in search of the food material, and we are throwing the food material along with the carry bag, plastic carry bag. So cow don't have the facility to open that carry bag and eat the food. So that's why along with the carry bag it will be consumed. Then this turtle is consuming the plastic which is suspended in the ocean. And here this particular right photograph is consisting of a whale. So whale is a organism which is existed in the deep of this solidified plastic that is being consumed by the fishes which are consuming the suspended plastic, that may be consuming the microplastics or even nanoplastic also. So this plastic is being consumed by the fishes, and after dissection you will find out the plastic material over there which are visible through our naked eye. But in addition to that, certain invisible plastics are also there. Because of that, we call it as microplastic. This will be only visible with the microscope. And nanoplastic, these are again invisible even for the microscope. So here there will be deposition of the plastic in the flesh of the fish or in the digestive system of the fish, and we can discover it, we can dissect it, and we can see this particular material. Next, please. So this plastic is not only harmful for the living organism or biota of the ocean, but this is harmful for the human being also. So what are the human impact of the plastic? We can see. Suppose the styrene is a polymer. This is considered as a potential human carcinogen. Suppose bisphenol A is there, then that is causing a diabetes, heart disease, or liver disease. And if these are phthalates, then these phthalates are the endocrine disruptors. So endocrine glands are there, and because of the endocrine glands, our body get regulated, and the reproductive system is also under the control of endocrine gland. So you know the story of the gonadotropic hormone. So they will be stimulating gonads and wait for the reproduction. So even such hormones are getting disturbed, and because of this, the reproductive problems are there. So just like the cases of PCOD or sterility, these are all because of this plastic consumption. Next, please. So these are the styrene. So styrene causes cancer, and these are the possible sources of the styrene. So foam which is made for the packaging or food containers or carpet or rubber or insulating material that is containing the styrene, and this is said to be harmful for the human being and it leads to cancer. Next. This is a BPA, bisphenol A. So bisphenol A is causing this particular problems. So changes in sperm morphology, decreased sperm concentration, total count, vitality, motility, immature sperm, changes in semen hydroxymethylation and miRNA, increased DNA sperm damage, changes in T, E2, LH, FSH, these are the names of hormones, prolactin, SHGB, and reduced Leydig cell functioning. This bisphenol A is related with reproductive system. Next, please. And here this is the story of phthalates. So phthalates, they are again acting as a mimicking substance for the hormone, and that's why the hormonal system are misinterpreted or misunderstood, and with this involvement of this plastic, this will be running out without involvement of the real hormone. So these havocs are there with respect to endocrinology. Next. Havoc or hazards due to what is our role, because it is invented by us and our duty to control it. So what are the possible control measures, how this is going to control, and what we should do for controlling the plastic waste that we have to understand. So very first is the restricted use of plastic. So as per the government rules and regulation, the plastic use is banned, and at every shopkeepers, if these inspectors are coming, then they try to hide the carry bags. Okay, they don't give you carry bag. But as soon as these inspecting people goes away, then again they start the packaging with this carry bag. Next, please. Next is recycling. So recycling is somewhat a useful strategy, because all the plastic polymers we can collect and we can sort it out and then go for the making of small bits by which new things can be produced, and this new thing may be utilized by human being. So this will be involving the process of fabrication, finally first the collection, then sorting, then shredding, then washing, and finally fabrication. This is a cyclic thing which we can implement during the can say recycling. So such recycling is possible up to certain extent. Next. Third is incineration, burning of plastic waste. If it is done, then this is going to release certain toxic gases. So suppose polyvinyl chloride, if it is taken as example, PVC, PVC if it is burned, then as it is vinyl chloride, so chlorine is getting released due to burning. So such type of toxic gases may be liberated, and plastic is basically hydrocarbon. So carbon dioxide or carbon monoxide, that will be obviously getting released. So carbon dioxide and carbon monoxide is harmful for human being, as it is combining with our hemoglobin just like oxygen, and carbon dioxide is a greenhouse gas. So this will be the causal agent for the global warming phenomenon. Next, please. Then another strategy is there that is known as landfilling. So where the material is not available, material means the building material, if it is not available, then instead of that plastic can be utilized. But of course it has certain limitation, and if the land which is filled with the plastic, if it is showing erosion, erosion may be caused because of the water or wind current. So due to this eroding activity, again there will be opening of the filled material. So there is a risk of this particular strategy. Then making road is again a very good strategy. It can be mixed with the tar or bitumen, and that form the homogeneous mixture. Homogeneous mixture can be utilized for making the road. So in major cities of India, this technology has been adopted, but of course it has limitations, and as this is hydrophobic in nature, so it is not binding properly, and whenever there are crevices, then this will be getting degraded after a particular time period. Then fuel generation is again one of the strategy. So most of the YouTube videos they are concentrating on this particular subject. So plastic material is taken in a container and this is supplied with the heat. Then petrol can be generated and that petrol is inflammable on another side. So for the making of this petrol, if you are spending lot of heat, then what is the use? Because heat is a fuel and petrol you are generating that is also a fuel. So there is no production is now recently get started. So biological material, if it is utilized for the production of plastic, then up to certain extent the problem can be solved. But the biological material is limited. Okay. So here the land is limited. Plants if you want to produce, then how much plant production will be there for the food and how much is to be targeted towards the plastic generation? That will be the question. Because land is limited, population is continuously growing, and with this growing population, the provision for the plastic or provision for the food material that will be again a question. And for this particular purpose, already existed land, medicinal plants, if these are there, these are present in the forest. So forests are to be eradicated, and then plastic growing plants we have to plant. Next. So degradation is a phenomenon which is slowly carried out, but surely it can be carried out. And this degradation is of so many types. So photo and thermo degradation these are taken as the central point in that. Photo means light and thermo means temperature. So light and temperature they are having the capacity to do work means the energy, and this energy are able to transfer the electron. Okay. So if the electrons are disturbed, electrons are transferred from this plastic material to somewhere else, then oxidation is said to be done. And when oxidized material is there, then the strength of that material is getting damaged. So up to certain extent, the degradation is possible by light and temperature. Next. Another alternative source is biodegradation. So here the biodegradation setup has been shown. Here the microbes are there, bacteria. These are going to secrete extracellular enzymes. Extracellular enzymes can work outside the cell. So they will be degrading the plastic material. And due to this degradation, the smaller water-soluble intermediates can be developed. These water-soluble intermediates can be taken up by the bacterial cell or microbial cell because they are acting as carbon source. So bacteria are dependent as a sole source of carbon on the plastic, then it will be okay. But bacteria, if they are having other alternative sources, then plastic degradation will be again in question. So this is the mechanism, possible mechanism of degradation of plastic by microbes. Next. So wetlands, we have scored. Why wetlands? Wetlands are said to be the best site for the waste management because it is having highest degradation rate. Why it is highest? Because there is a combination of land and water. And in water, both fresh water and marine water, these are getting covered. So mangrove wetlands, if you are considering, both the waters are available and both land and water these are available. So mud will be available and sometimes the air will also be inserted in that. So all the combinations are there which are helpful for the growth of microbes, and we have understood the capacity of microbes in an earlier slide. So wetland we can take as an example as appropriate site for the plastic degradation. So pertaining to this particular has focused on this to explore the wetlands and to find out the microbes which are having potential for the plastic degradation. And we have taken the example of plastic that is polythene. So experiences with polythene which we handled, I am going to share here with the slides onwards. So these were the wetlands which could scored by us. Next. This is the story of organic carbon. So as the time gets past, the organic carbon content is increasing, and microbes are most dependent on the carbon. So microbial load that will be also increased due to the accumulation of carbon. So as the carbon will increase, all the microbes will be increased, and obviously as the time passes, microbial population will be increased. And population of the microbes, if it is increasing, means diversity of the microbe is also ensured. So we can have greater biodiversity of the microbe because of the increased organic content. Next. So this is the decomposition mechanism of contaminants in wetland. Wetlands, if these are there, but what are the mechanisms? You can see here. Bioaccumulation is there. So waste material or contaminants are getting accumulated wherever these are there are biological material. So very typical example of say ionic interaction if we see. Bacterial cell wall is having negative charge, and positively charged ions are getting attracted and deposited on the negatively charged cell wall. So just like simple mechanism, the bioaccumulation works out. Then bioadsorption, again similar, adsorption and absorption. These are two words. These are well defined in chemistry. So inert material can adsorb the material like plastic. Biotransformation, because of the enzymatic setup, one particular polymer or one particular compound that is getting transformed into another format. Okay, information is there. Then next, biomineralization. Mineralization is whatever the minerals which are present existed, just like that, the minerals can be produced because of the biological activity. So suppose aerobic microbes, they are generating carbon dioxide. So that will be the example of biomineralization, because carbon dioxide is a mineral and that mineral is getting synthesized. Next. This is the plastic, and this plastic is being consumed by different mechanisms. So what possible mechanisms are there? First, the primary substrate. The primary substrate means the plastic is provided here as a sole source of energy. Next, the secondary substrate. Next means twice. So secondary substrate that provides the energy that is not available in high amount. And co-metabolic substrate means here the one compound is already taken up by the microbe. Along with that, if plastic is consumed, then that is known as co-metabolic, because both the metabolites are taken up. Next. So with this context, our work starts from this slide. So we could isolate the rhizosphere bacteria and rhizosphere fungi from the mangrove plants, and these are the sites we visited. 12 sites on the west coast of India. We visited and collected the rhizosphere material of the mangrove plants. So these are the soils from which rhizosphere material. Next, please. Degradation, if you want to check, then this is by which the first collection of the material. So rhizosphere of the mangrove we could select, and Avicennia marina, this particular species is found to be dominant. That's why we have targeted this, and the Avicennia marina plant from all the 12 sites we could select, and rhizosphere region that we have collected. Then afterward, the isolation of the microbes that was done. For the isolation, the LB agar medium and saffron agar medium that was used, LB for bacteria and Savarone for fungi. Afterward, the plastic material were inoculated with the broth media of this microbe, and then we have kept this in a regular shaking condition for the just say proper mixing of this plastic material and microbes or proper interaction of these two entities, and afterwards the screening was done. So for screening, the criteria were the weight loss and loss in the tensile strength that we have seen, and then confirmation we have done with the same scanning electron microscopy, FTIR, and finally the carbon dioxide evolution. So we'll see the results. So after screening of total bacteria and total fungi, these were the topmost. So Lysine Bacillus tuberculosis and Bacillus cereus, these two bacteria were found performing well for the degradation of polyethylene. And among fungi, the Aspergillus fluoreus and Aspergillus pseudomonas. So these were the isolates based on the two criteria. One is weight loss and another is loss in the tensile strength. Okay. So these are the same analysis. You can find the degradation. Here A is intact plastic, intact polythene, and here the cracks or scions or holes, all these are formed. So this is the indication of the degradation of the plastic. We have just scanned the surface of the plastic. Next please. Again, this is by fungi. In certain cases, fungal spores are also deposited. So in that case, due to the deposition of the spores, the criteria of weight loss that was not working, because due to the addition of weight of the fungal spores, the weight gain was there. So weight gain was again the indication of the degradation of the plastic that we could spore. And due to this dilemma, we could use another criteria, that is tensile strength. So that was also done. Along with that, the FTIR technique we have used gives us the value of carbonyl index. So carbonyl index, carbonyl is a group that is easily utilizable by the microbes, because its carbonyl is a C double bond O. So there is a formation of linkage with anything else, and that this carbonyl group can be utilized by the microbe. So carbonyl group formation, carbonyl index, that indicates that there is degradation. Next, here the FTIR by fungi, bacteria and fungi, both the results are there. Next. And finally, we could find out the carbon dioxide evolution test. So while doing this particular activity, the byproduct is getting formed by the microbe, that is generation of carbon dioxide. So this was also studied. In first case, there was no evolution of carbon dioxide, as the only medium is there, medium along with the plastic. But here in case of microbes, the evolution of carbon dioxide is there, and here there is a peak of carbon dioxide that we could find out. So these were the criteria by which we could confirm that the plastic is being degraded, that is degradable. Of course, the rate is not too much, and for this particular problem, we have to utilize the integrated strategy. So integrated strategy means all the combination of this technique we do, and if you sequentially arrange. So say for example, first the photo degradation, followed by thermo degradation, then followed by biodegradation, or rest of the plastic that will be sent to the recycling, or some of them which is utilizable that may be done for can filling or making mixture of the tar or bitumen and for plastic road preparation. So all these possible ways we have to think about, and accordingly we have to make a plan, we have to make suggest the strategies to the government, and we have to convince the government actually, then it will be applicable. So if we start thinking at our level, then that will be globalized, and the day will come, there will be plastic free environment, because today is the situation just like lithosphere, hydrosphere, atmosphere, there will be fourth layer that can be called as plasticosphere, that has occupied the whole earth, and therefore miserable situations we are facing. Next please. So with this, I am coming to an end. So I now acknowledge my students. Uh, Three of them they were awarded, and fourth one is working on this plastic degradation. So I am very much thankful to them, because of their contribution. We could run this particular mission towards the degradation of plastic or disposal of plastic. So I thank you very much. Next, please. So I have to thank first the microbes which are doing degradation, and of course all of you who are listening patiently from about 1 hour. So thank you very much, and I am especially thanking
Thank you so much, Professor Dr. Avinash Pillai, sir, for your enlightening and thoughtful, provoking incitement about your talk and about it is an astonishing talk as well as your work in the area of plastic management. And this is the actually demand of today's environment and the biodiversity to maintain our ecological conditions and the science very well. So this is really an astonishing work for our as well as we are very we are wishing you a great future ahead in this, and we can expecting that this environment is free from the plastic. Thank you once again sir for your amazing talk. Thank you so much. Yes. This session is open for question and answer. If anybody has a question, please ask.
any questions? I think hello, because this is the impact of question.
Sir, you have your
Sir, my question, sir. Sir, question. Sir, आता सध्या प्रिव्हियस रिसर्च मध्ये असं आढळून आले की जी ह्युमन बॉडी आहे ती ह्युमन बॉडीच्या प्रत्येक पार्ट्स मध्ये प्लास्टिक आढळून आले, नॅनो पार्टिकल्स. यावर तुमचं काय स्टँड आहे?
बरोबर आहे.
रिसर्च मध्ये आले.
Because we are saying that microplastic and nanoplastic. So nanomaterial can move from cell to cell. So if there are certain plastic bodies under electron microscopic photograph, then it is not surprising for us.
सर आणखी एक सांगायचं होतं की ब्रेस्ट मिल्क असतं ना, तर त्याच्यामध्ये पण आढळून आले. मग ते जनरेशन टू जनरेशन पास होऊ शकतं, कारण की बेबीसाठी मिल्क गरजेचं असतं.
Oh, obviously because microplastic, nanoplastic, if this is present in the soil, is through absorption, just like any other material that will be accumulated in the plant. Because hydroremediating plant, we know the cases. So this plant will be able to take the plastic. Because plastic is such material which mimics the other things. Okay. So plant get confused, which is to be absorbed and which is not to be absorbed. So that's why it will be obviously accumulated in the plants, and then this will be consumed by us. So we have to decide the tolerance limit, how much it is to be tolerated.
Good afternoon, sir. I am Rishikesh, Master Student of Molecular Biology and Biotechnology. Sir, first of all, thank you so much for the insightful and informative session. Sir, my question is while degrading the plastic, the organisms may also have some byproducts. One of the which was CO2. Is there any other byproducts which are more harmful than plastic?
We need to check. Okay, plastic that will be there obviously. There may be generation of oils. Okay. So oils, plastic is basically hydrocarbon. So we expect the oil generation. We expect certain, let's say, short fragments, oligo hydrocarbon, that kind of substances can be expected. So we have to go for more and more research, and we have to analyze those substances. Some of them we could analyze, but these are still to be tested on the organism for their toxicity test.
In your studies, you also find some byproducts. Others are yet to be discovered.
Yeah, byproduct is ultimate byproducts are two. In case of aerobic microbe, carbon dioxide. In case of anaerobic, methane. So these are the known byproducts. And obviously everything is present in the water, so water will be obviously byproduct.
Good afternoon, sir. Plastic waste management. We have two possible solutions. That is, firstly, utilize the available plastic and degrade it later. And second one is utilization of bioplastic. So, which one is efficient in your perspective? Please specify.
Bioplastic will be easier for the degradation, then I will prefer bioplastic. But as far as cost is concerned, that is more. And for generating bioplastic, we have to grow in lands, and land is limited. So that's why again we have to find out the intermediate solution.
Even durability is questionable.
Yeah. Yes, yes.
It is not durable.
No, it's not good.
At the same time, its tensile strength is also less.
Yeah.
Yeah.
We can't expect the performance like the plastic. So thank you very much.
Thank you so much, sir. Thank you. Thank you for the guidance and the now, our sessions chairperson, Dr. Rahul Anandway, sir, Head Department Mahavidyalaya Kutir, to express their chairperson of this session.
It's very great honor to chair a session with this intellectual session actually as a plastic waste management by a senior professor from Savitribai Pune University, Dr. Avinash RSML. Sir, as you told us, different spheres, and one of the plasticosphere at the last mention that you told. Really, we are still, the plastic is not replaced. A single-use plastic. I think more than 400 million tons of plastic throughout the entire day as a single-use plastic is to be consumed and utilized by the humans. So it's not a figure, it's not a data, but we are not having that much of ability to degrade it. So when the word comes to the management, Sir started as his lecture from as what's the matter, and that matter, as we know, the plastic. As different facets of the plastic waste management, he focused on the biodegradation. Though the different qualities of the plastic, that plastic is a very quite easily available. That's why a single-use plastic and so many different forms of the plastic that we are using daily. But it's our that a need of ours that we should focus on every researcher that in a line of that how Sir is conducting the research on the plastic biodegradation, how the insight is, particularly visiting the mangrove vegetation, that it is a very natural site, I think so, sir, because where the different types of rhizospheric microorganisms, they play a crucial role in the rhizospheric interaction with the soil where microorganisms, different types of plastics are to be there, garbage is there. Even the we called one terminology that I remember here, that is the toxic tidal waves that matters, and that is the very good place where we can focus on the particularly for the plastic biodegradation. Sir, enlighten with this lecture bit how the plastic production is increasing day by day, but it's an alarming situation, I think so. Plastic pollution as it has an impact on the humans and animals. One of the students of the post-graduation suggested that how from placenta to the bloodstream to the human milk and the breast milk, and even a very recent that nervous system, nervous coordination that we know that brain cells are also which have an impact of this plastic. So human a very greedy nature, and apart from that, we the whole budding scientists, we take this lecture as a very inspiring thought to us that we should focus on how to cure our mother nature, how to protect our mother nature from this plastic pollution. So everyone is even every scenario of the world, even so many scientists like RSML, they are working in this scenario to combat this pollution. At last, Sir has suggested that different species of bacteria, particularly fungal species, are very ubiquitously available. That is, a species of Aspergilla. They would lead to the very biodegradation process, which can be hastened. One more thing I add here, microbial population is very high. That number of microorganisms, they must be associated with that different bacteria and fungi. We must identify them. We go for the different isolation, identification, and at least we try here for the biodegradation. So, Sir has opened a very new chapter to us that we must work on that. Sir, you enlighten all of us. This is very new area of interest for the students also. Thank you very much, sir. Actually, you suggested it's a post-lunch session, but I observed, sir, everyone is very curious to listen how the plastic waste management through the biodegradation. And sir, you just open this as a very new chapter to us. Thank you very much. Thank you, sir.
Thank you so much, Professor, for your kind and guiding words towards the session. And I'm very grateful to you for your session words and talks for the inspirational work. It is really a motivational work. You have provoking our students for they can work on this topic, that is plastic management, as a new topic for their research. Thank you so much for being a chairperson as well as enlightening us, both of the resource person as well as the chairperson. Now, this is the time for the next technical session third. Thank you so much, sir. Now, moving ahead, we are honored by Dr. Rahul Chauhan, sir. Now I request we are award, sir, Botany Department, Rajarshi Shahu Mahavidyalaya, to express their thoughts of gratitude towards this resource person as well as chair person.
Good afternoon. I'm here to just complete the plans. And thank you to Dr. Rahul Alapurishan, Head Department of Botany, Udayagiri Mahavidyalaya, and resource person, Dr. Avinash Ade sir. He will enlighten us on plastic waste management with very much details. And ecology is one of the subject that is compulsory mandatory to all categories, like art, commerce, science, in their degree. So you should be study at least the pollution, and one of the major cause is the pollution of plastic. And that plastic how can degrade biological focuses in this subject lectures, and plastic can how recycle, reuse, and degrade, and how the mineralization process, decomposition process is focuses. And I also thank you to all the students here. Thank you.
Thank you so much, sir. Now, the third technical session has been started. Moving ahead, we are honored to have Dr. Rahul Chauhan, sir, an eminent researcher from Agricultural Biotechnology, Latur. I request you, sir, please come on the dais as a resource person. And now, I request the Dr. RSML, sir, Developnt Science College, Latur, to take a seat as a chairperson. RSML, sir, Developnt Science College, Latur, taking a seat as a chairperson of third technical session. Thank you, sir. Now I request to Professor Ashwin Shinde, sir, to welcome Department of Botany to welcome our session's chair, bouquet. Now I again request to the Hazare sir to welcome our resource person, eminent researcher, Professor Rahul Chauhan, sir. Thank you so much, sir. Thank you, sir. Now I request Ms. Neha Kamble to introduce our eminent resource person, Ms. Neha Kamble, for the introduction of a resource person.
Respected dignitaries on the dais and off the dais, faculty staff, and all my dear students, a very good afternoon to everyone. It is my great pleasure to introduce Rahul Chauhan sir, who has graciously joined us to share their expertise on the occasion of this national seminar. Rahul Chauhan sir is a resource person and he is a microbiologist and he did his master degree in agricultural biotechnology at Vilasrao Deshmukh College of Agricultural Biotechnology. He also did his PhD in biotechnology at SRTMU. He is an assistant professor. Rahul Chauhan sir also had so many awards in research papers like Best Research Paper Presentation in the National Symposium of Indian Society of Plant Pathology. He is also backed with Young Scientist Award from JMB Educational Society National Conference. He also awarded with Incentive Award publication by Department of Botany, Government of India in New Delhi. He also approaches detection, diagnosis, management of plant diseases here at Karnataka. He also approaches different memberships like of Indian Member Society of Plant Pathology at New Delhi, life members of Asian PG PR Society, expert member in IC Committee of College of Veterinary Udki. Dr. Rahul Chauhan sir handles different projects like some of the projects I would like to share with you. That is development of RNA mediated resistance in sunflower against sunflower necrosis virus. Plant tissue culture program for women angular development. Establishment of model genome clubs for sustainable agricultural development. I would like to share some special work of Dr. Rahul Chauhan sir, that is he developed XCM detection kit in cotton. We are honored by your presence on this occasion, sir. Thank you one and all, and I request Rahul Chauhan sir to communicate with all of us.
Now, welcome with a big round of applause, Dr. Rahul Chauhan on the topic research advancement molecular detection in the plant pathogens.
Thank you, one and all.
now it's good evening to all of you. I think everyone is tired. So I will try to make you interactive so that nobody will get tired. So my research topic is research advances in molecular detection of plant pathogens. So detection and diagnostics, so these are two different words. So everybody knows the importance of detection, plant pathogen detection or pathogen detection. So just 3 years back, we come out from the crisis of COVID pandemic. So everybody is realized the importance of the detection and early detection. What is the importance of the early detection? If we fail to detect like COVID-19, so this is a good example of early detection, and then how to implement the management practices, how to take qualified decision for the disease management. So the aspects I'm going to highlight the importance of disease detection, pathogen detections. What are the recent areas? So on which we can able to develop the technology and different techniques. Next thing. So, the importance of the pathogen detection. So, it is fundamental aspects of pathogen detections in even every field, including the clinical pathology. So, not only in agriculture, but in different things in medical field also, microbiology field also. It is very important to detect the plant pathogen. Then, second one is difficult to identify the pathogen because of their synergistic and antagonistic effects. Because most of the time, what happens? A patient or any plant showing the different symptoms at time, and we are getting confused. So, which is the pathogen? Which disease is there? So, it is very tough challenge, very difficult to identify the disease because of complex pathogens making this syndrome and synergistic effect, complementary effect. So, very difficult to identify such pathogens. So, in that cases, we have to find a tool so that we can efficiently identify the pathogens and take the proper management decision. Then enable to take qualified disease management decision at proper time so that we can avoid the epidemics. Then use of chemicals and precise and rapid detection of seed-borne pathogen. Because most of the time disease is not contagious, pathogen is not contagious. It may spread from the seed. It is present in a hidden form inside the seed, and it is acting as a carrier to transfer disease from one generation to the next generation. Sometimes what happens, disease is or pathogen is present in a hidden form. So this is called as a latent stage of infection. So morphologically we cannot able to visualize the pathogen, but still pathogen is there. So how to identify these pathogens and spread their productions and spread? Like seed is also one of them. Then we can able to detect the pathogen seeds and provide quality planting material, quality seed to farmers so that produce will be the quality. Next. So we can highlight some important diseases which made a huge impact on the agriculture research in the microbiology, pathology, and in prediction. So these are some diseases. Okay, we cannot forget such diseases. For example, Great Irish Potato Famine. So it is occurred in 1845. So this famine was due to a phytophthora infested pathogen. So this is the fungal pathogen. So the number of people get starved and died. Around 1 million people died because of starvation, because of entire food materials get spoiled and there is no food available. So 1 million people is the historical perspective. Next is for Indian scenario of the diseases and its importance. We can diseases in plant. So these diseases. These diseases puts the importance of the pathogen detections and their importance. For example, wheat rust, then red rot of sugarcane, Great Bengal famine. So 4 billion people's death reported due to this Great Bengal famine. Then wheat rust epidemics, bacterial leaf like of rice in the morning. Chairperson told bacterial leaf like of rice. So they have developed variety that is a sambal because of by the molecular marker assisted breeding. So this is a bacterial leaf like. Then tundra of rice, blast of rice, and banana bunch of virus in Tamil Nadu. So these diseases
may huge impact on the agricultural system and yield had been drastically reduced. Next. So these are the glimpses of the symptoms of this disease. Late blight of potato, then Great Bengal demise by Helminthosporium, then citrus cancers, and even recently, last decade, oily spot disease of pomegranate caused by Xanthomonas pudicii. So this is also a very devastating disease encountered in Maharashtra because of its loose pomegranate growing areas in Maharashtra. Next. So these are the viral diseases, yellow vein mosaic of the lady, then banana bunching virus, banana streak virus, then cocoa export virus, cotton liquid virus. These are the viral diseases transferred by the different vectors, and there is no resistance force available for such virus. We have to only proper management practices and restrict the entry of its vector for through early detections and very effective management practices. Only control such viral diseases. No resistance sources available for viruses. Next. So several methods are available. Conventional methods are available. So these conventional methods have certain drawbacks. So that's why we have to use some recent advancements and some molecular biology, molecular techniques for the detection of such plants. So time consuming, laborious and intensive. So this is the drawback of the conventional methods. Because we have to isolate the pathogen, we have to take samples, go into the laboratory, dissect it, go under microscope observations, and still we require expertise microbiologists to identify spore microscopic analysis. And some microbes they have a similar type of spores. Hundreds of the Alcalinae, hundreds of the Xanthomonas are there. They have a similar morphology. So it's very confusing how to identify. So then obligate parasite difficult to identify. Some viruses are there. So they are obligate parasites. They cannot multiply on the medium. So how to propagate such viruses? How to propagate such pathogens? And then how can we utilize in a observation purpose? So it's very difficult. Then closely related organisms difficult to discriminate morphologically because they have producing similar type of symptoms on the plant. They have a similar morphology. Then, so diagnosis of the plant becomes more difficult because of such limitations, and we need some sound practices and sound technology so that we can able to produce a quality planting material and for the better yield. Next. So why the molecular based technology for the pathogen detection? So because of the sensitivity, so they are very sensitive. They are highly specific. Even one seed is infected among the 100 seeds they can able to detect. Specificity is used. One pentagram of DNA, if it is present in a very minor quantity, they can able to detect. Then quantification. So these molecular based techniques not only able to detect pathogen but also able to quantify how much quantity of the pathogen present in a source we can able to detect. Then multiplexing approach and high throughput methods. At a time we can able to detect number of pathogens in one assay. So we can able to save our time, we can able to save our cost also because of such multiplexing and high throughput detection techniques. Next. So now these are the different approaches, molecular based detection system. So categorized into 5 to 6 different grades like serology based detection, ELISA based detection, deep sticks and lateral microscope based detection, then PCR based detection comes under the category of DNA based detection methods and DNA hybridization technology, then sequencing and then CRISPR technology. And another one is AI based detection of the plant pathogens, so latest technology. So we will highlight all these different techniques one by one. Next. Antigen and antibody detection. So this is the conventional detection. So precipitate ring test, gel diffusion test, elucidation test, and electron microscope serology. So this is based on the antigen and antibody reaction. So this is the earlier technology. If the antigen can able to bind to particular antibodies and then form it precipitation. So this precipitate can be observed in a tube or in a well, and formation of this spores. Based on this, we can able to find a particular pathogen if we bound to specific antibody and able to detect. Then next, this is the ELISA based enzyme. You might have heard this technology. So it is based on the antigen antibody reaction. So here there are two types of the antibodies: polyclonal antibody and monoclonal antibody. Then this ELISA assay based on the ELISA and Western body. So here antigen and antibody reaction is there. So this one is the direct antigen to the ELISA. So here we have to put antigen first on the ELISA titer plate, and then we have to add primary antibody. Then subsequently we have to add secondary antibody, and then secondary antibody is labeled with enzymes and antigen antibody complex is there, and substrate we utilize by these enzymes and form the colorimetric reactions, and then we can able to detect pathogen. Second one is double antibody sandwich method. So in double antibody sandwich method, we have to first immobilize our primary antibody on the ELISA or phytopattern plate, and then we have to add antigens, and this antigen bind to this particular antibody. Subsequently, we have to add secondary antibody which is labeled with enzyme, and then enzyme antibody reaction happens and it forms colorimetric reactions, and we can observe the color. So based on the presence of the color, we can able to find a pathogen is there. And intensity of the color indicated. So this is not only the qualitative method, but it is also a quantitative method. So it detects the presence of the pathogen and also they quantify the pathogen based on the amount of the color that is generated. So another one more important technique is deep sleep, or it is called as a lateral slow sleep method of detection. So this is a just instant method of detection of the pathogen. So based on this sleep, okay, this sleep is having a three compartment. So first one is a sample pad, second one is a conjugate pad, and third one is an absorbent pad. Okay, so sample pad, so that we have to make using so which we have to detect the pathogen. So this sample pad absorbs fast to capillary movement in upward directions. It conjugative pad, and in conjugative pad, so golden nanoparticles are immobilized, and subsequently it will pass through a test line. So in test line, there are antibodies which are immobilized. So antibodies are specific to particular antigen, and we bound to these antigens and follow colorimetric reaction. So if a particular color line is there, test line is there, and control line is there. Okay, both lines appears after the colorimetric reaction, then we can say the sample is positive. Particular pathogen is present. If the test line is present and control line is absent, then there is a spuriousness. Means our reaction is fault. So both lines should be there. And if the control line is present and test line is absent, then we can say our sample is negative. Next. So now based pathogen detection systems PCR variants are available for detection of the plant pathogens. So first one is the first generation PCR. So this is also called as conventional PCR. So PCR, here we have to use a pathogen specific primers. Primers will amplify a particular target from the genome and amplify a number of copies of target genes, and this can be detected on the gel of the system, and band should be there. So based on the presence of the band, we can able to find pathogen is present. So this is based on the just simple thermocyclic parameters or temperature cycle program is there. Second one is RT-PCR. So this one is called as reverse transcriptase PCR. So this is also called as bio-PCR. So this bio-PCR also not only detect the pathogen but also able to tell pathogen is live or dead. Okay, so two significant things are there. One is pathogen is present and pathogen is dead or alive. So that can be detected by this RT-PCR. Second one is multiplex PCR. So in multiplex PCR, we can detect number of pathogens in a single reaction. 5 to 6 or up to 10 pathogens we can able to detect simultaneously in a single set of reactions, so that we can avoid time. In case of the medical field, we can detect 10 diseases at a time, so that it is very easy and convenient for the doctor to implement the treatments within a time. Next one is SNP profile. Number of variants are generated, single nucleotide changes, it gives another new variant, is more dangerous infective. So such variants can be identified by the SNP profile, single nucleotide polymorphism profiles, based on the single base pair change. Then another one is nested PCR. So in nested PCR, for example, in case of the forensic science, if a particular sample is very rare, for example, hair fall. So hair fall is very less in quantity, and we have to use this for the detection of the victim, identification of the victim. So in that cases, DNA is very important part. So how to isolate DNA from very less samples? So in that case, we have to use this such nested PCR. So nested PCR has a two set of primers, inner primers and outer primers. So it will amplify use quantity of the target genes, and then gene specific primers will amplify the target gene from this rare samples and able to detect victim. So same way it can be utilized in plant pathogen detection. Next one is the MCH PCR, magnetic capture hybridization PCR. So in magnetic capture hybridization PCR, again, for example, microbiome, several thousands of microbes are present in a soil or any particular source. Rhizospheric soil, endospheric microbes, hydrospheric microbes. Okay. So from such microbes, we have to find a particular microbes from thousands of microbes by using such magnetic capture hybridization techniques. So it is based on the bead technology and probe based. So this probe will sort out the specific microbe that particular bead. Such a bead we have to collect and go for the PCR. Then PCR ELISA, so based on so two techniques are combined, PCR as well as ELISA. So first we have to amplify the target genes, and then we go for the quantification by using the ELISA, using the fluoro, by the recording the fluorescence. Then real-time PCR. So it is also called as RT-PCR. So everybody is familiar with RT-PCR because in COVID we have used commonly such RT-PCR test. Everybody has gone through this test. So it is a real-time PCR test. Real-time is what? It gives a real presence of the pathogens, not only presence but real quantification. How much quantity of the virus, how much quantity of the pathogen present into the human bodies or in plant bodies. So that can be measured based on the tissue cycle. So in RT-PCR or in real-time PCR, we are familiar with score. What is the score of RT-PCR? If it is 30 and more than 30 and below than 30, if it is CT value, then patient is infected. If it is more than so such type of the for the quarantine detection of the pathogen. Then another recent technique is LAMP and RPA, that is recombinant polymerase amplification. So here we didn't require PCR machine. Without PCR machine, we can use the same technology for detection of the plant pathogens. Recombinant polymerase amplification. Recombinant polymerase amplification, it has three enzymes. One is DNA polymerase, second one is recombinant polymerase, and third one is single strand specific. So these three enzymes bind together and bind the target sequence, and at the same temperature, around 37 to 45 degrees Celsius temperature, unique temperature we require. Nothing is required, no PCR is required, only a hybridized water bath is required, because we have to maintain the single temperatures and be able to amplify, detect the pathogens. So it is a handy portable, you can able to detect pathogens at on site. So these are the important target genes. Generally, housekeeping genes, mitochondrial genes being used for the detection, because COX gene in mitochondria, mitochondrial COX gene in case of the plant, because these genes having a high evolutionary rate, so that able to find any evolution, any changes into the particular pathogens, and we can able to trace it. Next. So this is the PCR-based detection. It is based on the principle of denaturation, annealing, and extension. Three-step procedure. Double strand get denatures, form a single strand. Then particular primer get aligned with particular complementary regions, and with the help of back polymerase and DLTP, it will extend the amplification of the target regions, and we will get a multiple copies of the genes which can be able to trace under a agarose gel or gel documentation system, and then you can find the presence of the pathogens. Next. So this one is the lab-based detection. Lab-based detection, that is a loop-mediated amplification. So here in case of the lab, so we have to use a 6 set of timers and 8 regions being targeted. So this 6 set of timers along with the lab timers, so it will combine to each other and it will amplify with particular thermostable enzyme that is called as BST polymerase enzyme. So these BST polymerase enzymes will amplify at specific temperature 60 to 65 degrees Celsius. So here also we don't require thermocyclers. So in water bath we can do these reactions, and so that is the beauty of this technology without a huge costing. In simple water bath at single temperature 60 or 65 degrees Celsius, with the help of the enzymes you can able to detect the presence of the pathogen. So this is the colorimetric assay. So based on this reaction, again amplification, based on the color formation, we can able to find pathogen is present or absent. If color is not formed, then pathogen is absent. Next. So this is a recombinant polymerase amplification based on, again, this, it is also called as RPA based detection. So in this RPA based detections, again don't require thermocycler. Temperature reaction is only a single step, 37 to 42 degrees Celsius temperature. Recombinant polymerase enzyme is required. Then back polymerase enzyme is required, and single strand binding specific proteins are required. So it will dissociate and two strands simultaneously being amplified by the recombinant polymerase enzyme at 37 degrees Celsius. So this is also the techniques where we can use at on-site. On-site means particular location on field without using any costly equipment. Next. So this technique can be able to detect 1 to 10 copies of the DNA. If it is present in very rare quantity, so we can able to detect. So these are the different techniques. So now these are the different molecular diagnostic assays we have developed in our agricultural important crops. Oilseed crops, particularly in sunflower, we developed the detection marker for the Alternaria disease. Then Alternaria longipes, Alternaria diantricola, Alternaria alternata, and a test australiensis. So these are the different fungal pathogens able to cause Alternaria blight disease in sunflowers. So we developed molecular marker system for detection of such pathogens. Then cotton, this is the important fiber crop, and it is being infected by the bacterial blight. It is also called as angular leaf blight disease in cotton. It is caused by the pathogen Xanthomonas campestris, or it is also called as Xanthomonas malvaceum. So we developed a marker for the detection of such a bacterial blight pathogen of the cotton. Then in case of the onion, we developed marker for the Alternaria forae. Then in case of the fruits or pomegranate, and in case of the banana, we developed a diagnostics assay for detection of this Xanthomonas pudicii pathogen. Next. So now this is the technology we developed. So this one is the VNMQV alpha detect. So this technology is based on the real-time PCR-based assay. So we developed a marker and probes which are specific to this Alternaria LAC disease. So based on this Alternaria LAC, we designed probes which are specific to this particular LAC and primers which are being amplified and which are overlapping to the probe. And based on these principles, we have developed a ready-made assay. So that assay just we have to take and add the pathogen sample which is being used for the testing. and run the reaction. So after 1.5 hours, you can able to detect the presence of the pathogen. So leaf sample, seed and spores. You can use this source for the detections, and this technique is able to detect one seed. If it is one seed is infected from the 100 seed, you can able to detect with the help of this technology. And just recently we got a patent for this technology. Next. So another is banana bunching virus. So it is caused by the DNA virus in banana. So almost everybody is familiar with banana. Generally, we are using tissue culture grown banana plants, and tissue culture grown banana plants should be virus free. Before sold to the farmers, that should be tested with the help of such a virus if it is present or absent. So this is called as virus indexing. So every tissue culture protected materials being certified with the help of such a virus kits or virus indexing technologies. So banana bunching virus is the most important virus in banana. We have to certify it whether virus is present or absent, and if it is negative, then we can able to sold, we can able to sell this banana plant, this tissue culture plantets to the farmers. So this is also real-time PCR based detection. So we developed a assay for the virus detections. Just we have to take a matrix sample from the plant, banana tissue culture plant, just dissolve in extraction purpose and use as a template in the assay, and within a 60 minutes you can able to detect whether virus is present or absent. Okay, so this is also a very handy technique. Next. So this BBT detection is based on the Tagman probe based technology. So this Tagman probe based technology is here we have to use a specific probe. So this probe is labeled with a reporting tag, that is fat or depth, and another 3' we have to label it with the quencher. So the probe is hybridized to particular regions, and so that tag polymerase dissociate from 5' to 3' end, and if probe is dissociated from particular targets, then it quenches and reporting time both are dissociated from this probe, and then it emits the fluorescence. So number of times it is being amplified with PCR reaction. So number of times it is dissociated from particular strands and it emits the fluorescence. So if the number of cycles are there, more dye is accumulated inside the reactions, and this dye being captured by the real-time PCR. And this fluorescence, amount of fluorescence generated during the reaction, that can be measured with the help of fluorometer or real-time PCR, and it gives the CT value, and based on the CT value we can able to find pathogen is present or absent. So this one is the technology. So this is the BBTB assay. So we developed this diagnostic kit for the detection of the banana bunch virus. So this is also got patent in last year. Next. Another molecular detection of the bacterial blight disease in cotton. It is also called as angular leaf spot disease. So this is caused by the Xanthomas unique or Xanthomas malvaceae. So here we developed a conventional PCR based detection assay, and this assay is able to detect even this pathogen, presence of the pathogen from the infected tissue, from bacterial pure cultures, and from soil. So based on this amplification, we can able to detect the pathogen is present, and then we can able to implement a chemical control strategy if it is present into the sample. Next. So this one is it. So in 2016, we got patent for this technology. So this is the XCM detection kit for the bacterial blight detection in cotton. Next. Then DNA fingerprinting, another most important approach, DNA fingerprinting. So DNA fingerprinting means we are getting a different banding profile. So banding profile of every species, every human beings, every pathogen is unique. It is based on the number of genes which are available, type of genes which are available, and based on the amplification patterns. Every pattern is different. So based on these patterns, we can able to find a particular pathogens, because a pattern, DNA fingerprinting pattern is unique for every species or every human being. And we can use such markers, that is RFID marker, SSR marker, ISSR marker, an SNP marker for detection as well as differentiation of every pathogens within the group and within the species also. Next. So this is the DNA fingerprinting profile. So this is the unique profile for particular species, and based on this profile, we can differentiate the pathogens intra as well as interspecies level also. Next. So another one more technique, that is the DNA based hybridization based. So here DNA hybridization techniques. So we have to use a DNA as a probe, and this probe will bind to a particular region on the genome, and based on their binding capabilities and hybridizations, and but later on we can able to detect it on the basis of the chemofluorescence technology or X-ray field we can detect, and then we can say the pathogen is present. So this is DNA extraction followed by restriction dilation followed by electrophoresis. Subsequently, we have to transfer in nylon membrane, cytocellular membranes. So this is called as dotting. Then subsequently again transfer on the nitrocellular membrane, then hybridize with probe, and then detections, and last we will get a particular image. So based on this image, we can able to find the pathogen presence or absent. So by using this hybridization based technology, so we developed a race specific marker for detection of the bacterial bite disease in water. So in water there are number of races are there. 19 races are there. So race 18 is a very destructive race of the bacterial bite disease. So based we have collected different samples from the different locations, and we have got genomic enzymes and then hybridized with ABR gene as a probe, and based on their hybridization patterns, we found a particular two fragments which are common in race 18. So this is the particular fragment of 40 kb and 6 kb. These are common in a race 18. So based on this pattern, unique patterns, we can find that. So this is the race 18 marker for identification of the race 18 of Xanthomas malvacearum. Next. Again, DNA microarray technology. So here, microarray technology. So it is has a tremendous applications. It is not only used for the detection of the pathogen, but also it is being used in gene expression study, presence of the gene, identification of the different cells. So here, probes are immobilized on the particular plate. So it is called as glass plate, and this probe will hybridize into sample source, and we can find such type of these spots. So such as spots are appear, so we can categorize even live cells are there, dead cells are there, particular type of the cells are there. So based on this, we can able to find a particular gene, and based on such data, we can also able to find a particular pathogen. Next. So another technique is sequencing. So this is also a latest technique. So in case of the COVID situation, we can also identify different variants within a short span because of the sequencing. Okay, first variant, second variant, third variant. Third variant is more aggressive, so that all people are okay. So threat generated among the people because of such a variant. So how to identify such a variants? So such variants are there. So variants can be identified by using the massive sequencing technology. Number of sequencing platforms are available. So such platforms like NGS technology, then Solexia, Illumina sequencing. So all automated sequencing. So within a short span of time, we can generate huge data over the genome of particular pathogen, and we can able to categorize, we can able to analyze with the help of the bioinformatics tools and find the variants. Next. So next one is DNA barcoding. So DNA barcoding. So we all are familiar with barcodes because barcodes are available everywhere. If we go to purchase from the clothes, also there is barcode. Okay, so barcode is a unique identity for particular things. So same barcodes are available for the pathogen or any plant species also. So these barcodes are generated based on the unique sequences of particular genes. So number of genes, for example, mitochondrial genes are used. Then ribosomal DNA, internal transcript special regions genes. So they are used as a barcode genes. So these sequence. So you can see this is the population. Population. subsequently we can use the DNA markers and differentiate all the populations of microbes. And this population, with the help of such barcodes, we can categorize and we can find a genetic relationship among all these pathogens. Interspecies and other intraspecies. Okay, find the genetic distance among each and individual pathogens with the help of such barcodes. So now CRISPR, okay. So since morning we are discussing about CRISPR. So CRISPR is a cluster based short palindromic repeat sequences. So this generally CRISPR is used for the genome editing to edit the particular genes, develop mutants, engineer the microbes. But here CRISPR also being used for the detection of the plant pathogens. We have used Cas12a proteins assemble with different RPA or lab variant technology and then use two. So here application of the CRISPR is gene functional studies by using gene knock-in and gene knockout. Gene knock-in is we can add particular coded genes and allow to express. Gene knockout is what? To silent technique. Okay, then microbes detection. So CRISPR also used for the detection of the microbes. Then engineering of microbial communities. Okay, suppose if we want to increase the performance, increase the activity of particular microbe, for example, biodegradation of the plastic, we can increase its activity by engineering such a microbes, targeting specific genes, increase its enzymatic activity, and then we can use such an engineered microbe for the biodegradation, pesticide degradation, contaminated sites, that is called as rejuvenation of the contaminated sites, soils. So we can use such engineered microbes with the help of the CRISPR-based technology. So CRISPR is not only used for the genome editing, but also used for the detection. Next. So this is the CRISPR-assisted detection of the plant microbes. Just recently published in paper in virology. So DNA virus and RNA virus. So both we can detect on site with the help of CRISPR-based technology. So we have to use a particular probe. So that probe labeled with reporting time and quencher time. Use RPA-based primers assembled together with Cas12 proteins, and this RPA will amplify target genes and CRISPR Cas12a will edit it. If it edit a particular probe which is bound to particular regions, fluorescence is generated. And fluorescence is generated, that fluorescence can be reported with the help of spectrophotometer or fluorometer. So here we did not require particular instrument. We don't require PCR, we don't require any electrophoresis system. So directly we can just a single fluorometer is required to read the or to measure the absorbance generated during the reaction. So this is an on-site technology. On field we can able to detect the pathogen. Next. So now this is the AI and artificial intelligence and IoT based detection of the plant pathogen. So here, so this is the flow chart. So number of steps are involved how to use AI technology for the detection of the plant pathogen. So this is the database of the different crops. So it requires huge database. Number of images are required from single stage specific. Okay, aging, infection, severe infection, maturity, full number of images are required. We require a huge database. Then image acquisition. So we require high image capturing devices, high resolution devices for the image capturing. We require satellites so that image can be captured from every angle. Then image processing, processors are required. Then we have to able to analyze the data, and here based on the analysis we can able to detect, we can predict or we can propose the model. Okay, we can propose the forecast whether disease appearance are there or disease epidemics will be there. So such type of the assumption we can do with the help of such AI and IoT based detection or technology. Next. So now coming to the last point, conclusion. So molecular diagnostic technology requires to move from qualitative and quantitative because not able to detect presence or absence of the pathogen, but also we can able to find how much quantity of particular bacteria, particular enzyme, or particular virus is present in given sample, so that we can do a particular management decision. Disease management decision. Then pathogen diagnosis and quantification to estimate potential risk regarding disease development. So based on the AI technology, IoT technology, we can able to forecast, we can predict whether in future coming days disease favorable condition is there or disease may be formed in the form of epidemics. So then what should be the strategies to be indicated for control of such diseases? So we should be alert for this system. Then such diagnostic system, such predictions at molecular level can force us or can make us to take efficient, take qualified decision for the disease management. Then it is useful in production of the quality plastic material. So not only it is able to control the diseases, their spread, but at early stage, whatever the agricultural produce are there, quality planting seeds are there, new agricultural produce. So that should be free from diseases for the human consumption. So next. More emphasis is required to increase detection specificity and sensitivity, because if your technology is not sound, not able to trace the minimum quantity of the pathogens, not able to trace a particular pathogens from the huge population, then there is no use of the technology. So technology should be a sensitive, it should be a specific. So more research innovations are required to make our technology sensitive and specific. Then multiplexing and cost effective. Okay, number of pathogens can be detected simultaneously, and we have to reduce the cost of the technology. Okay, if particular virus can be detected in 1,000 rupees, so within same cost, we can detect 10 viruses. So ultimately, cost will be down. So we have to propose a technology advancement in multiplexing ways. Then requirement of the field requires portable rapid assays. Detection in laboratory is very easy, but we have to bring the technology to the field. So make it convenient to the farmer, make it convenient to the end user. Then our technology will be popular. So research invention should be in such way that bring it from lab to lab. Then extensive data sets are required, training with diverse samples and environmental conditions and negative parameters. So, based on the AI and IoT technology, huge data sets are required, number of huge processors are required to analyze the images, number of models, algorithms are required so that we can propose the models and find out the technology in a sustainable way. Then required advances in sensors, automations and robotic technology. Now this is the era of mechanizations. Automation, so everything should be in an instant. So in such way that our technology should be move rapidly. So it should be automatic. It should be a sound. It is based on the sensor. Based on the sensor, we can able to detect the pathogen. So this is the technology advancement. So in such way, we should come forwards and do the research so that based on the biosensor, just scanning, if the device seed materials can be scanned and should be generated in a fraction of second. So such type of the invention should be required in future generation. And I think this is last. So I acknowledge to the resources and our students. So we are going to work under such aspects as MSc students, PhD students, and we develop such a technology for the detection of the plant pathogens from different crops. Thank you.
Thank you so much, sir. Thank you so much for your insightful session and your presentation and engaging the interaction. I request to student this session is open for question and answer. If anybody has a question, you can ask him.
I think everyone is tired.
No, sir. Our students are very, very enthusiastic. They are not tired at all.
We are enjoying those.
Your explanation is so depth and enjoyable.
As day by day, new technologies are evolved, and come forward for purpose is nice. So, sir, is there any nanotechnological approach for detection of plant pathogens in practice?
Yes, there are nanotech based some biosensors are available. There are combination of the biosensors and nanosensors. Okay, so by using these combinations, so several techniques are available for the detection of the pathogens. Even some aptamers are there. So aptamers and in combination with this nanotechnology, so we can able to find take pesticide residues in this food material.
One more question.
There is the term of plant nanobiologics. Is there a role in plant pathogen detection?
Which one? Plant
Plant nanobiologics.
biomix,
Bionanobics.
plant nano
Biologics.
biomix. Okay. So so this plant nano biomix, correct? So this plant nanobiotics, so there is a some plant microbes, nanomaterials. So this plant nanobiotics, so again biomix is prepared from such microbes. Okay, so it is maybe endosymbionts and maybe some rhizospheric microbes are there, and maybe some trichodermas are there. So nanomaterial is developed from such microbes, so it is being used for a different purpose as a bioagents and stimulating the growth of the plant, taking care of diseases, pesticides. So it is being used.
Is there anyone? Thank you so much, sir, once again for your time and effort, valuable contributions. Now I request our chairperson for expressing the views about the session and conclusion of the session.
Good afternoon, everybody. So all of you are my ability, very try, but still, few minutes will be left. This is for first time I am saying certain session. So if any mistake by me, please excuse me. Chavan sir had delivered very nice lecture on the recent advances related to the biomarkers, which will be used for the purpose of detection of plant pathogens. So we know that traditional methods to detect the plant pathology or plant pathogens they are there, which are considered as field detection as well as lab diagnosis. Now this lab diagnosis and field detection, it was also earlier it was not available, as sir have mentioned a disease which have caused a very big famine called as iris famine. So this iris famine, it was happened during 1840s and it was continued for 15 years. And due to that famine, as sir already have mentioned that about 10 lakh people or 1 million people they have died of starvation. And more than that, means about 20 to 30 lakh people they have migrated or they have left Ireland. They went to America. Today's America was mostly occupied by the Irish people as well as Australia. And that famine, why that famine was caused? And high potato crop was very poor in those days. That was not known to those people. So it was many commissions as well as committees have been founded by their parliament to find out the cause of loss of potato. But still, there were 14 years later, in 1876, we have found out that it was a fungi, a single small microorganism, who is responsible for death of 10 lakh people and migration of about 30 lakh people from Europe itself. So nowadays, better techniques they were now available for the identification of diseases. But still, there is my own question. See, every species have its own limit, which is set by the nature. So, I think humans have crossed their limit. And as humans are crossing their population limit to survive on the earth, they are taking resources of some other animals as well as microorganisms. And what we are doing? We are killing pathogens, means microorganisms. as well as we are killing insects, we are killing many other pests, just like rats, snakes, etc., etc. And their resources we are utilizing to fulfill our human stomach, or we say human needs. So somewhere I think we have to stop, so that I think we should give right to other organisms or other species to survive there also. So this is the session. It was very better session. So I think I will stop here. Thank you all.
Thank you, Anand. Thanks for the final. Thank you so much. Now,
Thank you, sir. Thank you very much. Now I request the winner of this seminar. Friends, see, some of you do not give patience to listen what work these researchers did, and you don't know the mystery of the experiments. The mystery of the experiments requires a lot of patience. You have to perform the n number of experiments, and not necessarily each experiment will give the result, but you keep patience, and you are not having the patience to listen this word. So keep patience. Now I request convener of this seminar, Professor S.N. Shinde sir, to felicitate the chairman of technical session first, Dr. Rahul Alapore, with our tradition and the tradition of our college, by offering a memento and a book. Dr. Rahul Alapore, sir, please.
सर थोड़ा सीनियर
I also request once again Professor S.N. Shinde sir to felicitate Chairman of Technical Session Second, Professor Dr. Anil sir, by offering a memento and a book. Thank you.
now I request Ms. Neha. Vote of thanks for this session. Technical session third. Ms. Neha Nagre for the vote of.
As we come to the conclusion of this insightful and enriching session, it is my honor to deliver the vote of thanks. First and foremost, I extend our deepest gratitude to our resource person, Dr. Rahul Chaman sir. Thank you, sir, for your presentation, which are truly valuable and thought-provoking. Your valuable insights have significantly contributed to the success of this event and have provided us with much to reflect upon. I also thank to our chairperson, a heartfelt thanks to our distinguished speakers and panelists for sharing your expertise and knowledge. Your presentation and discussions have been incredibly informative, engaging, offering us a new perspectives and ideas. A special thanks to our participants and attendees for your active engagement and enthusiasm. Your presence and contribution have enriched this discussion and made this session a collaborative success. To the technical support team, thank you for ensuring that our session ran seamlessly. Lastly, I would like to thank our venue staff for their excellent service and hospitality. Once again, thank you once again for your valuable contributions. You made this session a memorable and impactful experience. Have a great evening, everyone, and all.
Thank you so much, Pam. Thank you so much. Now this is the time for the next session. Next